The effector functions of T cells are determined by the array of effector molecules they produce. The effector molecules produced by armed effector T cells fall into two broad classes: cytotoxins, which are stored in specialized lytic granules and released by cytotoxic CD8 T cells, and cytokines and related membrane-associated proteins, which are synthesized de novo by all effector T cells. The cytotoxins are the principal effector molecules of cytotoxic T cells. Their release, in particular, must be tightly regulated as they are not specific. They can penetrate the lipid bilayer and trigger an intrinsic death program in any cell. By contrast, cytokines and membrane-associated proteins act by binding to specific receptors on the target cell. Cytokines and membrane-associated proteins are the principal mediators of CD4 T cell effector actions, and the main effector actions of CD4 cells are therefore directed at specialized cells that express receptors for these proteins. The membrane-associated effector molecules are all structurally related to tumor necrosis factor, TNF, and the receptors on target cells are members of the TNF receptor family. All three classes of effector T cell express one or more members of the TNF family upon recognizing their specific antigen on the target cell. The membrane-bound TNF family member, CD40 ligand, is of particular importance for CD4 T cell effector function. It is induced on the Th1 and Th2 cells and delivers activating signals to B cells and macrophages through the TNFR protein CD40. TNF-alpha is made by Th1 cells, some Th2 cells, and by cytotoxic T cells in soluble and membrane-associated forms, and can also deliver activating signals to macrophages. Some members of the TNF family can stimulate death by apoptosis. Thus, FAS ligand, CD95L, the principal membrane-associated TNF-related molecule expressed by cytotoxic T cells, can trigger death by apoptosis in target cells bearing the receptor protein FAS CD95. Some Th1 cells also express FAS ligand and can kill FAS-bearing cells with which they interact. Death by this mechanism appears to be important for removing activated FAS-bearing lymphocytes. If it fails, a lymphoproliferative disease associated with severe autoimmunity results. And that's the effector functions of T cells are determined by the array of effector molecules they produce. Have a great day.